Monday in our research symposium series. Last week we had a number of biology folks talking about some of their work, and today we're going to talk a little bit about chemistry and physics. Our first speaker today is Alex. He's going to be speaking about sound and how we perceive speech. We are also going to hear from a couple of chemistry folks who are going to talk about their work on understanding how proteins bind small molecules. Eujin will be talking about how tuberculosis puts together a coat that makes it hard for an antibiotic to get at, and in particular how it might be possible to make some molecules for the formation of that coat. And then we're going to hear a little bit about how cannabinoid receptors bind small molecules and the effects of things. And so without further ado, please go ahead.
Hey guys, my name is Alex Radulescu, and over this semester I built a text-to-speech engine. So we're taking text, specifically formatted for the engine, and it's going to output what should sound like human text. So for my example, I'm going to make it say, hello friends, but for the input text, I have to type it exactly as you see here. So it doesn't sound exactly like a human speaking. It's a little bit robotic. But I think the theory behind it is really interesting. So I'm going to talk about how I got to work, what isn't working yet, and I still have to fix to make it sound better. And then last I'll talk about other cool stuff I can do now that I have this concept. So here's a diagram of a sound wave. On the right side you have a sound source, something that's vibrating. Every time it vibrates, it sends out a pressure wave that travels out longitudinally. So as it approaches a human ear, we would perceive this change in pressure as a sound. As long as it's over 20 hertz, that's our minimum frequency that we need. So instead of showing it like that, we show it as amplitude versus time. And this is the most basic sound wave. It's a sine wave. So I'm going to play a sine wave for you guys that's 200 hertz. And this is going to be the bass I'm going to compare for you guys. Okay. So there's four characteristics of sound waves that I was changing for this experiment. I was changing the frequency. So I'll change it from 200 to 300 for you guys. And you can hear that when I change the frequency, it changes the pitch. It makes it sound higher or lower. Then I'm going to change the amplitude. So this is the original sound. And this is me playing it multiplied by 0.5. And you can hear that it's a little bit quieter this time. So amplitude pertains to volume. I can change the shift, and it sounds exactly the same after I do. And then last, I can change the shape of the wave. So from a sine wave to something like a sawtooth. So that last one, the shape, pertains to the timbre of the sound. That's the difference between a piano playing a note and then a violin playing the same exact note. It's the same frequency, it's the same amplitude, but we can still tell the difference. And that was especially important to me for this project because the same difference happens between the ah and the o when someone talks. So I had to figure out what shape the human makes when they say certain syllables. And at first I decided maybe I can just take the waveform, copy it, and then paste it when I need it. So if someone wants the ah sound, I'll just play myself sing it back. But there ends up being a couple problems with it. So here's a couple of ways to show the shape of a wave. One way to show the shape of a wave is through a Fourier transform. You can run this function, say a basic sine wave through a Fourier transform, and then what you get out is a vectogram. And this one in particular has a peak at 200, and it's at amplitude 1. So it shows that this frequency, this function is made up of one sine wave at frequency 200. Now it gives another one, and the 200 sine wave added to a 300 sine wave. So I ran that one through the same Fourier transform, and I ended up with a spectrum with peak at 200 and peak at 300. So all the Fourier transform does is show you what sine waves you have to add to get the function back. And this works for every function. This is a box square wave, and I ran through the spectrogram is a little bit more crazy looking. And then this is a sawtooth, the same thing. So what got me curious was that the spectrogram shows the amplitude and shows the frequency, but it doesn't show a shift, which is a little confusing because if you change the shape, the entire shape of the wave changes. So wouldn't that change the way it sounds? So I have a sawtooth here, and I put it through a Fourier transform, and I got this spectrogram. And I wrote a function in MATLAB to generate every single sine wave that pertains to that spectrogram and add them manually one by one. And for this diagram, I did it with no shift. And you see it's the same exact shape, it just happens to be flipped backwards. But here I did this one with a slight shift. For every frequency, it shifted over by some small fraction and starts being distorted. I did it for an even bigger fraction this next time, and the shape's fully rounded up. And then for the last one, I added random shift to every sine wave I added. But what was crazy was that for every sound, for each of these sound waves, if you played it, it sounds exactly like the original sawtooth wave. So that showed me that the first idea was a terrible idea. I could just record myself saying ah and then play it back. I wouldn't understand anything about the wave. Instead, I needed to take a spectrogram, look at what frequencies were dominant and what amplitudes they were at, and then plug that back in. So here's me doing that for the ah valve. So I measured it with a spectrogram that looked like this, and you can see three peaks. There's one here, here, and about there. These peaks are called formants, and they're the dominant frequencies of the valve, and they're different for every single valve. It's the fingerprint of the valve. So for I saw the formants around 800, 1100, and 2500. And I wrote a function specifically for that follows the shape of a normal distribution curve that I can use to put frequencies into and see what amplitude they should be. So from this measured version of I got this generated version that should have no noise and be stronger in the places where it should, because I made sure the formants were amplified. Now, for another kind of sounds, fricatives, that's things without tones like s and f. It was very similar with one difference. Here, if you look at the vowel, you can see that there's these gaps between each frequency. Those gaps are the same as the value of the fundamental. So that means that when you would listen to the sound, you would hear it at that pitch. But now this doesn't have a fundamental, it doesn't have a pitch. So all I had to do was make sure that I added every sine wave in between the harmonics, and I would still get back saying the sound like so. So I'm going to play my engine saying ah. Now I'm going to play it saying so. So it still doesn't sound perfect, right? But you can tell the difference. You can tell what sound is trying to make at least. So now what I still need to fix. This is a spectrograph of someone saying a sound and it's measured. And you can see a couple of things here that I don't account for. So for example, this is someone saying tan. And you can see that these bands here are frequencies that are the formants. And they shift for some reason. So when I was writing this engine, I didn't account for that. But somehow when you say these sounds, they change based on how long you've been saying them. So when I refine this engine, I'm going to have to find some way to account for that. And once I do, I'll also be able to have this solved, a consonant. This was a death to me. I couldn't figure out consonants because here you can see there's total silence. This is where you hold back the air, and then here you release it, t. And that's totally time-based, and I just couldn't figure out how to do it. So if you listen to me running a text-to-speech that's on my system, if I remember what I'll call it.
Hello.
So you can tell that even when it says the O, it shifts this frequency down. It doesn't say it says O.
No.
So once I refine this, it will be able to mimic this more closely. And finally, now that I have these concepts down, what else I can do? What else can I do? So one thing I tried was actually learning backwards. That ended up very easy because I have all the functions written, right? So I can record myself saying something, as long as it's only one sound, one syllable, and I can have it spit out in text form what the syllable was. And you're going to take my word for it because I can't show my screen to you guys. And then another thing I was interested in doing, I didn't get a chance to do, was solving instruments, the same way I solved vowels. Because I solved something like A or O just by taking a spectrogram, just find what the formants are, and I play it back, and I can get that vowel sound at any pitch. So I was curious if I could do that to an instrument. So next I want to try, say, maybe recording a violin and seeing if I can change the pitch and have it still sound like a violin. And that's it. Thanks for having me. Special thanks to Professor Mina because she listened to my first 100 takes, so you guys don't have to. So I take questions.
Any questions for Alex?
Hi everyone, my name is Eujin Lim. I'm a chemistry and biology major right now in Middlesex, and my current research is searching for a viable inhibitor for Mycobacterium Tuberculosis. The initial start from this research, I was actually originally researching electrochemistry at the moment, but then during one of Dr. Chris's PowerPoint, she actually mentioned mycolic acid, attributing to why Mycobacterium tuberculosis can be very tedious to deal with. And then after talking to Dr. Gosha, he recommended me to research further into this topic. This is when I encountered this article published in 2015 by Eujin Lim. The title of the article is called "An Enzyme Essential for Mycolic Acid Biosynthesis." And the important thing to note about this is that it shows the known crystallized structure of the specific enzyme. And the article stated that the enzyme stated in this article, called FAD32, is essential for this biosynthesis of mycolic acid. And so to give a brief overview of what MTB or Mycobacterium tuberculosis is that MTB is essentially an airborne bacilli. In addition to that, it's an obligate aerobe. What that essentially means is that it can only be contracted through the contact air, such as like breathing or coughing onto someone else. In addition to this of being an obligate aerobe, it means that it requires a high amount of oxygen in an outside environment in order for it to grow. That's why typically when doctors tend to do X-ray scans for TB, you'll notice that it's commonly bound in the lungs or sometimes in your bloodstream. And the way it infects the host is that it actually intentionally lets itself get consumed by the macrophages that's commonly found in the lungs in the image shown right here with the blue being the macrophages. And the unusual thing about the Mycobacterium tuberculosis is that the mycolic acid present prevents the lysosome found in the macrophages from actually destroying the bacteria. So it's essentially just living inside the macrophages. And the problem with this is that it has a thing called a virulence factor, which essentially means that as more white blood cells or macrophages continue to clump around the Mycobacterium tuberculosis, it eventually forms this solid surface called a granuloma, which is typically white. And once the Mycobacterium tuberculosis decides to activate, it will then cause tissue degradation, also known as commonly known as necrosis, and then it will continue to infect other parts of the body, which eventually leads to death. And many people wonder how severe actually is tuberculosis. According to the World Health Organization report from last year, approximately 1.4 million people actually die from tuberculosis alone from worldwide, and it's actually considered the 9th deadliest disease in the world. And other things that contribute to its deadly effect is that it always continues to start developing new antibiotic-resistant factors, which prevents first-line drugs such as rifamycin and isoniazid from actually working. And to develop a new drug to actually fight these kind of specific kind of Mycobacterium tuberculosis, it can actually cost anywhere from $115 to $240 million, sometimes even greater. And like I stated earlier about the article I stated earlier, the enzyme I want to focus on is an enzyme specific called FAD32. And the function of this enzyme is to essentially activate a substrate called meromycolic acid, which then is condensed, which gives the mycolic acid its potent properties. And a couple months later, I then discovered a new article by Misty L. Kuhn that uses the same FAD32 enzyme, but rather than using the substrate ATP, it uses a ligand that's shown in yellow called ligand 649. And the green image presented right here shows the active site of the residue in which the ligand is interacting with. And the interesting to note is that the article specifically stated in red that it can be a promising drug target for treating tuberculosis. And the idea kind of interested and motivated me to further research this topic. So after talking to Dr. Ghosh about this topic, he soon then gave me the full approval to just go on with the research. And now I realize that the goal of my research is to design a new inhibitor for this FAD32 enzyme. And that if the design ligand is successful, then it could potentially be further researched so that it can potentially be synthesized as a drug. And the approach that I use for this research is to use molecular docking to help identify inhibitors for this enzyme. And then there are certain protocols that I would need, such as I will need 3D structure of both the FAD32 enzyme and the 3D structure of the ligand. And in order to see these structures, I will also need a visualization software to see the specific interactions. And then in order to dock the molecules to get the result, I will also need a docking software. And in the next slide presented, these are the 4 softwares I use for my research. And then on the top left right here is called the Protein Data Bank, which is essentially where you can find various virus enzyme structures or bacteria structures or any other human enzyme structures. For my case, that I decided to search up my FAD32 enzyme. Once I got the structure, I then used UCF Chimera, which is a visualization software, which is also assisted with docking, to see the crystallized structure of the specific enzyme. After I was able to see the structure of the enzyme, I'll then proceed to start developing my ligand, first by using Chem2D draw, and then start minimizing the structure using Chem3D draw. After my enzyme structure and my ligands are completed, then I would then start proceeding to use SwissDock to start docking the results and see what I get back. And in this image shown right here is showing how I was able to dug up the FAD32 enzyme structure. By going onto the PDB website, I just searched up the FAD32 enzyme. And as you can see right here that's circled in red, there's 4 different FAD32 structures available. And the image shown right here after is just one of the 4 results that I got. And the image right here that's been boxed is the 4 ID code needed in order to see the enzyme structure in Chimera. And then the image shown right here, with the use of Chimera, I designed two FAD32 enzymes, but on the right shown right here, rather than, I used the ligand ATP, which is shown in the sphere right here, and then on the colorful image on the outside is to show the beta sheets and the alpha helices in the enzyme. And then on the left right here, again, I decided to use the ligand 649, which was mentioned earlier on the article shown. And then instead of using the ATP, I decided to use ligand 649. And then also, on the next slide shown, I decided to superimpose the 2 images shown. That, as you can see, that the ligand 649, colored in purple, stretches toward the left, toward the hydrophobic pocket. And then on the right, shown on the ATP, with the triphosphate heading toward the other direction. The interesting thing to note is that the adenine structure of both the 649 and the ATP, including the ribose, superimpose on top of each other. So things to note is that, number one, this is an unusually very large pocket for this specific enzyme. In addition, there's one hydrophobic pocket shown right here due to the long carbon chain, and there's a charge region due to the triphosphate. And then the point of this is that to essentially, I want to reproduce the same result, that I'm going to dock the same enzyme structure and use the same ligand 649 ATP. The purpose is this, that to establish my proof of concept is to show that if my results are very similar to the known crystallized structure results, then I'm sure that my crystallized structure can be used to dock the unknown molecules to see that if it can inhibit my FAD32. And then on the next slide is to show how I was able to dock my results to establish my proof of concept. The first slide, text box right here is to show the Swiss doc website, which is showing where I would dock the results. And the first thing I need to do is to prepare my protein structure for Chimera. The procedure for this is essentially to remove any non-essential items that might be presented inside ligands, such as like ATP or any water or any structures like that. In addition, there is also decide to work on also defining my binding site / center, which can be found using Chimera, in addition using the PDB website. After my protein structure is then completed, I will then proceed to start developing my ligand using ChemDraw right here on the image shown. After minimizing the structure, I then start placing each file according to what is required. Then I will proceed then to start submitting my docking job, and then through roughly around overnight, you will then receive an email of what you're saying your job is now terminated. In this case, for the next slide shown, I decided to show my docking results in regards of ATP. And then the results shown right here is typically what you see when you get your results back. And they're typically split into a couple of categories. In this case, they're split first into clusters and element, which is essentially showing the location of where it was docked, in addition to how the ligand is placed. And then it was categorized by delta G. And delta G basically refers to Gibbs free energy. It shows the energy released from the enzyme. And then when noticing about the delta G, the important thing to note is that generally as the negative value of delta G increases, usually the binding affinity will increase as well regarding the protein and the ligand interactions. But in this case, I'm just trying to match my results as close as I can with the known crystallized structure. Hence the reason why I didn't choose the higher delta G value. In this case, on the next slide I will show, I chose cluster 0 element 5 with a delta G value of negative 10.37. And then the image shown right here, on the left first, it shows the known crystallized interactions of the FAD32 enzyme with ATP. And on the right, you can see right here is that the blue represents my dock results of ATP, and the ball and stick that's also shown in purple represents the known crystallized interaction. The thing to know is that the adenine structure and the ribose structure superimpose on top of each other, and it shows nearly a 90% similar accuracy. That it is understandable, though, for the triphosphate that's shown right here from mine, that it would not identically match with the triphosphate shown right here with the crystallized structure, mainly because the triphosphate is a flexible molecule and is expected because my Swiss dock cannot dock the magnesium that's present in the article stated, because magnesium is used to help stabilize the crystallized structure. And then with the next image shown, I decided then also to dock the ligand 649 because I also need to further establish my proof of concept. And in this case, again, I just need to choose one ligand one result that nearly matches with the known crystallized structure. And then the dock results shown right here, again, blue representing mine, and then the purple ball stick representing the crystallized structure. And my structure nearly had a 95% similarity, that although only because of mine so, that it shows nearly a 95% similarity with all the interactions shown. And in the image shown right here is to now show that I actually proven my proof of concept. In the image shown right here, that as you can see, that my ligand 649 nearly matches with the known structure right here. And the image shown is to essentially just show a cavity image of what the enzyme of the ligand will look like inside the specific FAD32 enzyme. With this known, now I could then proceed to start docking unknown molecules for this specific enzyme. The one thing to note is that Dr. Ghosh actually wrote the specific article with these following ligands. And interesting to note that when talking to Dr. Go, she actually recommended me using these specific molecules, because when comparing with ATP, it has very similar molecular structure, including the ribose sugar structure right here, and similarly with the phosphate. The only difference is just the guanine structure rather than the adenine structure. And he thought that because this structure is originally used as an antiviral agent, then maybe there's a potential chance that this ligand can also work with FAD32 enzyme. So when doing my first doc results, the first one I decided to start with is the 7-methylguanosine triphosphate. When comparing with ATP, the ribose structure and the phosphate structure is actually identical with the ATP. Only the difference is that rather than the adenine, it's actually a guanine structure that's replaced alongside with a charged nitrogen and methyl group. And in this case, when choosing the result for which one we best known, I decided to choose the one that's almost similarly to adenosine triphosphate, which has a value of negative 13.14. And then when superimposing the two images shown right here, blue again representing my doc results and green representing the ATP shown, that the guanine structure and the ATP structure are nearly on the same plane but with different interactions, and that they actually are similarly in the same regions. But again, the thing to note is that the triphosphate, rather than heading straight, is actually heading toward up. But again, it's noted that the triphosphate is very flexible, and this is the interaction that it feels like it was the best suited for. And the thing to note is that the value for the delta G for my 7-methylguanosine triphosphate is actually higher than the adenosine triphosphate, which is interesting to note, is that this molecule that's used as an antiviral agent actually has a higher value than adenosine triphosphate alone. And then now when docking a second molecule, I decided now to use 7-benzylguanosine monophosphate. And in this case, rather than using methyl group, I use a benzene group alongside taking off 2 of the phosphate. It is a thing to note that the delta G value is slightly smaller than the 7-benzylguanosine triphosphate, but I'll explain that in the next slide. And then the image shown right here is essentially how the ligand will bind inside the pocket. Now, when showing the dock interaction right here, again, the blue represents mine and green is crystallized structure. I decided to actually use the ligand 649 as my reference structure because of the fact this benzyl ring that's present right here. This benzyl ring actually has a similar hydrophobic interaction similarly with the ligand 649, as you can see that's entering the pocket. And the thing to know is that the pocket exists right here is where the meromycolic acid would enter inside the enzyme for the final condensation. And in addition, the guanine structure, rather than being where the adenine structure is right here in the middle, and it has a T shape alongside with the ribose with this hydrogen bond and alongside with the phosphate with its other bond. Although the structure for the green is higher in terms of delta G, it is an important thing to note is that the 7-benzylguanosine monophosphate is actually a lot smaller than this ligand 649. So in this case, that if I decide to improve the structure, maybe extending the benzyl carbon chain right further, then there might be a possibility of a higher delta G increase. And the thing to note is that because the molecule is small, there are further methods of improvements when it comes to using this structure. So the conclusion I was able to come up with is that, number one, I was able to successfully adopt some of Dr. Ghosh's molecule, and that the 7-benzylguanosine monophosphate shown earlier on the previous slide can be shown to have improvements to help improve the delta G value. And in addition to that, is that there was a certain issue when regarding to docking, specifically the structure called 4EI1. When docking this molecule, it actually showed an error called a topology error, which is essentially just a 3-dimensional structure error when it comes to Swiss dock. And then now, the end of the day when it comes to this is that due to computer modeling, that this could suggest that this molecule could be a viable lead so that the molecule can be actually further synthesized into a drug to help treat people with TB. And then now I would like to acknowledge everyone that helped me with their presentation. I would like to especially thank Dr. Ghosh and Dr. Sukta Ghosh with the presentation. I would actually like to personally thank Dr. Sukta Ghosh with assisting me with PDB and Chimera. And I'd also like to thank Dr. Levy with his criticism. And I'd also like to give a special thank you for Dr. Rowley giving his insight to my presentation, helping improving it. In addition, I'd like to thank Dr. Christensen for actually providing me with the idea, because it actually came from just one of her slides in the PowerPoint. And then lastly, I'd like to thank the Dean, Linda Scher, and then Dr. Parag Muley and Dr. Michael Ansonoff for providing this opportunity to do this research. In addition, I'd like to thank the lab coordinator, David Rosen, all my peers, and then I'd like to thank Bristol Myers Squibb for their financial report. Thank you for listening to my report, and I'll be happy to take any of your questions.
I would just like to give a comment that research is really interesting and it could lead to discovery of a drug for TB. Have you considered collaborating with a pharmaceutical company in the future?
Currently, no, because I'm a student. But currently right now is that the doc results shown earlier. Sorry, this battery died. The doc shown earlier is essentially just showing the binding affinity. It doesn't guarantee that this molecule will directly inhibit it. Normally, in this case, I would use Swiss prediction, but the problem with using Swiss prediction is that my bacteria specifically is not. My FAD32 enzyme shown earlier is that is not available for Swiss prediction, so I cannot accurately definitively say that it will inhibit the enzyme. I'm just giving the possibility in which, based on the delta G binding affinity value, comparing a ligand 649 ATP, that there is a possibility it can work.
Basically, we would like to do that one, but the point is we do not know that this thing is going to bind that effectively infect the pocket. That's our result. It's a theoretical result. Now, of course, I would like to move forward because this drug is already been active as an antiviral, active compound.
That lays the foundation for
Right.
further research.
He just laid out the foundation on this.
Yes, the next step would be to do a biological assay, taking that enzyme and testing it on.
Okay, thank you.
All right, how's everyone doing today? I said, how's everyone doing today? Yeah, that's what I like to hear. My name is Josh Alvin. For my research project this semester, I conducted it on cannabinoids, cannabis, and the Endocannabinoid System. Now, I know most of you know or heard of cannabis, pot, and marijuana out there, but, you know, there's been some research done over the past 50 years since its discovery by Dr. Raphael Michelin, but, you know, some people don't really know how it interacts. And even though we know that it treats many different diseases and ailments, there still are some questions left as to how exactly it does that. So my research analyzes this. You see, when you ingest cannabis, it interacts with what is known as the Endocannabinoid System, which is a system of endogenous receptors and lipids that assists in the regulation of homeostasis. Now, there are two types of cannabinoid receptors that are currently known. The CB1 receptor, which is located on the central nervous system, primarily in the brain and reproductive organs, as you can see here, and the CB2 receptor, which is located on immune-related tissues and peripheral organs, as you can see here, the digestive system. Now, the endocannabinoid system interacts specifically with what are known as cannabinoids, all right, which are any molecule that interacts with cannabinoid receptors. These are the molecules that are most commonly associated with the active components in cannabis, okay? In cannabinoids, they can function in one of three different ways: agonist, inverse agonist, or antagonist. And you can think of those ways similarly as to operating a vehicle. When you ingest an agonist, it takes the vehicle, puts that car in drive, right? You're going to go forward with this. It's a chemical that binds to the receptor to produce a biological response. The most wide-known agonist that's a cannabinoid is THC, tetrahydrocannabinol. This is what's in your joint when you're smoking. THC is most common, what has been shown to treat nausea in cancer patients, and, as most of you know, induces the munchies. Yeah, that's what makes you raid your fridge late at night. An inverse agonist is the opposite of an agonist. It takes that car, puts it in reverse, okay? It produces the opposite biological response. The most wide-known inverse agonist is rimonabant, okay? And unlike THC, it makes you less hungry. Look at this guy. He doesn't want any food. He's like, nah, put that away from me. And an antagonist, okay? This is going to take your car, put it in neutral. No matter how hard you hit that gas, you're not going anywhere, okay? The most wide-known antagonist is cannabidiol. Again, it's inhibiting any other response with the molecule. It's blocking its effect, okay? CBD has been shown to treat anxiety, like Charlie Brown, you know, and has been shown to treat grand mal epileptic seizures in children, which is what it's most popularly used for. This Jaden Davis, this patient that the company I used to work for treated, and as you can see, his father is administering him a CBD tincture. Now, these cannabinoids interact specifically with the CB1 receptor. Again, it is a cell membrane receptor located throughout the central nervous system, and is responsible for things such as the sleep cycle, appetite, mood, and body temperature. This is an actual image of the CB1 receptor. As you can see, it's shaped similarly to a boot, okay? And when this is THC docked within the receptor. When any molecule enters, it comes through the calf into the back of the toe and docks right where, and through the back of the foot and docks right where the toe is, putting itself, placing itself like Cinderella putting on that glass slipper. Now, how do we actually obtain this? Well, this leads to our protocol, putting the pieces together now. Thanks to Dr. Raymond Stevens from the University of Northeastern, the first crystal receptor, this first crystallized CB1 receptor was actually done on the Antagonist 5TG site. Okay, in 2016 it was published in the Cell paper. And now, as recently as July 2017, was the Agonist 5XR8 CB1 crystallized site published in Nature. Okay, these are the same exact CB1 receptors, okay? As you can see, there's an antagonist crystal structure here with the surface, that pocket where it's actually placed in the receptor, and here is an agonist surface. Again, that pocket which is in the receptor. Same CB1 receptor, but when you superimpose the two, you can clearly see how they're slightly different, okay? Which shows that depending on the type of cannabinoid you are ingesting will depend on the different conformation of the calf of the boot of the CB1 receptor. As you can see, here in blue is the agonist, and in pink is the antagonist, and it shifts. It wobbles, the receptor changes, meaning there's differences, meaning there's something else there. So, although many small molecules have been tested in the CB1 receptor, okay, and we know that agonist and antagonist molecules bind to the same CB1 pocket, but induce different conformations, can we distinguish between agonists and antagonists based off of what amino acids they contact? Now, the purpose of this is to determine what the binding conformations of CB1 antagonists and antagonists are. We will do this by observing which amino acid residues they contact, and we will conduct this through using a series of bioinformatics computer modulating softwares for the CB1 structure. The tools we will use will follow a similar protocol to my associate Eugin. Okay, we will obtain the CB1 sites from the protein data bank here, and then we will make the cannabinoids themselves using Chem2D and Chem3D draw. Use those cannabinoids, plug them into the Swiss target prediction, which will tell us our probability and affinity to bind to the CB1 receptor, and to plug it into the Swiss docking program, which will give us our different ligand conformations within the protein. And then we will view our findings here in the UCSF Chimera. we reported the data here. These are the 8 cannabinoids I've decided to choose to send in for the experiment. What type they are, in other words, how do they function, the proper site of function, the size of the pocket measured in angstroms, and the center of the pocket. These X, Y, Z coordinates are exactly where I am putting these cannabinoids in the receptor. And then the IC50, we will then compare the IC50 value, inhibition constant. In other words, how much of this given cannabinoid or ligand does it take to reduce 50% of the activity within the CB1 receptor, and then compare it to the unknown delta G value measured in kilocalories per mole. That delta G value is going to tell us how much energy is being released based off of the different poses we see within the receptor. Again, the more negative a value for the delta G, the better binding. That's what it equates to. I know for most of us who are taking Gen Chem 2, we learned that delta G, that gives free energy, is measured in kilojoules. This is in kilocalories. Again, you should take this number and multiply it by 4.81. So, after getting all this information together, where do we start? Well, I started with the first one that was published in 2016, the Antagonist site. These are the 4 cannabinoids I chose. First being ZDG. Okay, this is the crystallized structure in the receptor, and this is going to be my proof of concept. Then I moved on to the inverse agonist we talked about, called rimonabant. After rimonabant, I moved on to the synthetic cannabinoid CB1 antagonist taranabant. And then, again, the most widely known phytocannabinoid antagonist, cannabidiol, or CBD for short. So ZDG's actual name is AN6538. That's how it's better known. Again, the synthetic cannabinoid CB1 antagonist. The Swiss target prediction report, this is the bioinformatics tool, again, I used. And this is a 2D image of ZDG. This is a full prediction report stating how the cannabinoid receptor is a high probability to bind to it. So it looks pretty good so far. And these are the results from the Swiss docking program. On our left here, you can see this is just another image of the CB1 receptor. And right here is ZDG, that ligand inserted here. And on our right, we get our different protein conformations here that are measured, that again, are based off their measurement in the kilocalories per mole that gives free energy here, that delta G value, okay? Again, the more negative, the better binding. I selected the ones that had the best correlating pose to the crystal structure, similar to how my friend Eujin did. So this is the best dock pose I was able to generate for ZDG. Okay, as you can see, these are the residues it interacts with. All right, it's a hydrophobic interaction with the pocket, meaning it's holding itself together in space. And it forms this Y shape that you see. Okay? When we examine the crystal structure pose, again, that's this similar Y shape. Now, when I superimpose the two, you can see how they both look very similar. There's a 90% similarity of my dock pose to the corresponding crystal structure and how they sit perfectly within the same pocket, meaning that my protocol is correct, which gives us confidence moving forward to the unknown antagonist that I sent in for docking. So let's see. I moved on to rimonabant next. Again, that synthetic CB1 inverse agonist we talked about. Here's the Swiss target prediction report showing high probability to bind to that CB1 site. This is the best dock pose I was able to generate. And as you can see, it forms this Y shape. Again, interacting in the hydrophobic pocket. And then, when superimposed over the crystal structure, you can see how, again, same residue interactions and similar Y shape, about 90% similarities. There's a couple differences here with these rings that may be oriented a little differently. But again, 90% similar to the corresponding crystal structure. Okay? If you can't see from there, here's a better angle. Again, forming that Y shape that we keep seeing. Afterwards, moved on to taranabant, the synthetic cannabinoid CB1 antagonist. As you can see from the Swiss target prediction report, it has high probability to bind to that CB1 site. And interacts with the same residues that we see Y shape. Now, when superimposed over the crystal structure, you can see again similar Y shape and same residues. You can't make it out from that image. A better one is here, showing how same Y shape and the same Y pocket, about 90% similar to the corresponding crystal structure again. Lastly, cannabidiol, or CBD, the phytocannabinoid CB1 antagonist. Phytocannabinoid, all that means is cannabinoid produced in the cannabis plant. That's all you have to remember. The Swiss target prediction report, this was the best target prediction report actually. The CB1 receptor, highest probability to bind to it. Look at that, it's full green, just like the plant. Now, CBD, the best docking pose, it forms this Y shape. There are a few less residues it interacts with, but again, it interacts with less residues because it's a smaller molecule. But again, similar residue interaction, and the best dock pose we were able to generate. When superimposed over the crystal structure, you can see here, again, same residues, again, forming this Y shape that we keep seeing. Slight differences here, but maybe better images here floating in space. As you can see, Y shape. Difference here, instead of a ring, it has this pentyl chain. Now, this Y shape, after this study, we can conclude, I don't know, some of you might know that there's a difference when ingesting THC to CBD, and how when you ingest CBD before ingesting THC, how you do not get the same effects. This explains that. Because CBD forms a Y shape, that Y acts as a blockade. That blockade prevents THC from interacting with the CB1 receptor, which is why you do not have the same effects, which is why you will not have that generated psychoactive effect. This explains that. Our results from here, as you can see, our delta G values are all similar except for CBDs. CBDs is less by about 1, and the reason for that is because it's a smaller molecule. Again, smaller molecule is going to have less residue interaction, meaning the delta G is going to be slightly less. And compared to the IC50 values, as you can see, these two match up. Tyranobans and cannabidiol CBDs match up. Because tyranobans is a larger molecule, means it'll have better binding, means less IC50 value. And CBD, again, slightly smaller, so it'll have a higher IC50 value. And these are the key residues. Again, all hydrophobic interactions that occur within this pocket. Now, after concluding this information, I decided to move on to the 2017 Nature paper for the Antagonist site. And I first started with this cannabinoid here, 8D0. This is the crystal structured cannabinoid that's in that CB1 site. And then I moved on to anandamide, dubbed AEA. Anandamide, or AEA, is the cannabinoid that's an endogenous cannabinoid. In other words, it's the cannabinoid your body naturally produces. I don't know if any of you are fitness freaks like I kind of am, but whenever you experience that runner's high, this is what you're experiencing. Okay, it's this cannabinoid interacting with that CB1 site that gives you that generated feeling and effect. Now, moved on afterwards to cannabinol, or dubbed CBN for short. Okay, it's actually a derivative of THC. And then afterwards moved on to THC itself. So 8D0, synthetic cannabinoid CB1 agonist, as you can see from the Swiss target prediction report, has high probability to bind to that CB1 site. This is the dot structure we were able to generate. As you can see, there's a hydrogen bond that occurs between this alcohol group in red and sarinine 383, this residue here, okay? And the rest of the residues it interacts with are all strictly hydrophobic. A better image that can be shown here is this aerial view. As you can see, it forms this L shape, okay? Which is important to note when we go through the rest of the experiment. Results. This is the crystal structure that's shown. As you can see, again, this hydrogen bond is right there. Again, the rest of the residue interaction is strictly hydrophobic. And an aerial view can be seen here. Again, that same L shape as shown before. But when the two are superimposed, you can see how the rings match up slightly. There's some differences here and here, and again, that hydrogen bond is still there. A better view can be seen with this aerial view, forming that similar L shape you keep seeing. And when put in the pocket, you can see how the crystal structure is in black, and my dot results are in blue. It's kind of hard to make out, but again, in that same pocket interacting with the same residues, there's about over 90% similarity to the corresponding crystal structure, meaning that my protocol is correct, which gives us confidence when going into the unknown results. Anandamide, AEA, an endogenous cannabinoid CB1 agonist. As you can see, it has high probability to bind to that CB1 site, which is expected. Again, it's the cannabinoid your body makes specifically to target that site. This is the best doc pose we were able to generate. It interacts with a few more residues that aren't shown here, but this is just for presentation purposes. So you can see it's this long, greasy chain. There isn't any hydrogen bond that occurs here. That's because there's no alcohol group. Again, it's just a long, greasy chain. When superimposed over the crystal structure, as you can see, it forms similarly. It goes in that same pocket, and a better view can be seen from this, interacting again with the same residues in a similar pocket, and again, right there, 90% similarity to the corresponding crystal structure, meaning this is correct. Next, I moved on to cannabinol, CBN, again, the phytocannabinoid CB1 agonist. As you can see, it has high probability to bind to that CB1 site. This actually had the best results for the agonist when binding to the CB1 receptor site. As you can see, this is the best doc pose I was able to generate. Again, there is a hydrogen bond within here that's not shown, that you can see here. So it is within hydrogen bond distance. Again, it just wasn't calculated or shown. And this aerial view, as you can see, it forms a similar L shape within the pocket, meaning it is over 90% correct to the corresponding crystal structure. Now, lastly, THC, tetrahydrocannabinol. This is Snoop Dogg's favorite molecule. Okay? This has high probability to bind to the CB1 receptor. Yeah. Okay, that should be obvious. Now, the best doc pose I was able to generate. As you can see here, there's no hydrogen bond that occurs between sarinine 383 and this alcohol group because the ring is pointing away. Sarinine's here, the ring's pointed this way. Okay, it puts it out of hydrogen bond distance and reaches a strictly hydrophobic interaction with the pocket. There is a unique residue that THC interacts with that the rest of the cannabinoids don't. Lysine 192, right here. Okay? Now that's going to be important. Again, the only residue that THC interacts with the rest of them don't, which explains the profound psychoactive effect. When superimposed over the crystal structure, you can see how, again, this is the crystal structure in black, THC is in blue. It doesn't exactly form the same way. THC, the rings don't line up exactly. It doesn't make this L shape that you can see here from the aerial view. It goes straight, where this kind of bends to the right. The results here, as you can see, the delta G values for AP0 and anandamide, AEA, are similar, and THC and CBN are similar. Why? Well, with AP0 and anandamide, they're longer, greasier chains. What did we say before? More molecule, more negative delta G value. THC and CBN are very similar to one another. So of course they're going to have a similar delta G value. And these are the key residues they interact with in this area. So, what can we conclude from all of this? Well, after compiling the list of seeing all the residues antagonists interact with and all the residues agonists interact with from this, I was able to see which ones are the same and which ones are different. So the agonist and antagonist residues that are the same are shown in green. But in pink are the ones specifically antagonists, all the antagonists interact with in the 5-TGZ site, and in blue are all of the agonist residues that the agonists interact with in the 5-XRA site. And in red here is the specific residue THC interacts with. This is important to note, because this basically defines what an agonist is and what an antagonist is. This will help us determine now how to build synthetic cannabinoids to target these specific residues, and how to build agonist-bound cannabinoids to target these specific residues. In conclusion, computer modeling of the known compounds match the X-ray structure. After examining the binding of antagonists and agonists, we can successfully conclude that the way agonists and antagonists bind are different and predictable. I'd like to acknowledge my research advisors, Dr. Ghosh, Dr. Brian Levy, and Dr. Sutapa Ghosh, for helping me throughout this thing. If it wasn't for them, I wouldn't have been able to do this at all. You know, Dr. Ghosh is the one that worked with me since August of 2016, giving me the confidence to get into this program. Dr. Brian Levy's worked in the industry with synthetic cannabinoids for 4 years. I studied them for over 10, and with his knowledge and oversight, I was able to conduct this successfully. And Dr. Sutapa Ghosh was able to teach me how to use the protein data bank and Chimera program successfully to conduct this experiment. I'd like to thank the financial support for Bristol Myers Squibb. Special thanks to Dr. Stephen Riley for his help and insights. Dr. Linda Scherz, the Dean of Natural Sciences, and Dr. Parag Muli, the Chair, and Dr. Michael Anselov, the Associate Chair of the Natural Sciences, for letting us have this course. Dr. Lab Coordinator Dave Rosen for his help. Eujin Lim, my only other partner in crime with this, and motivation, encouragement, and assistance. All of my peers for putting up with me throughout this entire thing. But then, Dr. Albert Poles II, the CEO and founder of CBD Science LLC. He's the one that mentored me in cannabinoids, and I worked in industry for 2 years. And my father, Dr. Edgar Alp, DMD, for supporting me throughout all my endeavors over these past few years. I love you, Dad. And I'll end on this. It is not about saying no, N-O. It's about saying no, K-N-O-W. Know what you are ingesting and how it affects your system, and only then will you truly be able to make wiser decisions when consuming any substance. Thank you very much. Any questions? Doctor?
So I was looking at what you were saying here. So let's say you end up, I come to you with a completely novel chemical.
Okay.
I say it interacts with the cannabinoid receptor, but I have no idea if it's an antagonist or an agonist. How would you use the methodology you developed to be able to give me an idea on which one it's going to be?
That's a great question. So we would look to input it into the 2 different sites. It's actually important to note that when conducting this experiment, I did take the antagonist cannabinoids at first and I put them in the agonist site. And what I noticed is that they weren't lining up right. There were 2 CBD molecules that were in the superimposed over, and the stuff wasn't lining up right. But when I took the antagonist cannabinoids and then put them in the antagonist site, bang! It was like peanut butter and jelly, just matched perfectly. You know, so through conducting the series of steps that we did in order to find the key residues, in order to see how and why each one bound to the specific pose and how it's superimposed compared to the crystal structure, that will be able to give us the answer that you're seeking. Any other questions? Any questions regarding the cannabis, cannabinoids, anything like that? Anything you guys want to know? No?
Is it legal to buy and possess these cannabinoids for research purposes?
That's a great question. Okay, well, there is, if you have a Schedule I DEA license that only a few schools have, primarily the University of Mississippi, Ole Miss, has the only license to cultivate cannabis on site, then you'll be able to use these for research purposes, and you can actually contact them, and as long as you have a DEA license to work with these specific cannabinoids. There's the medical industry that is legal out here. They do not actually make any type of tinctures or edibles, nothing like that. It's just straight plant material, which is detrimental to its consumers because it just enables them to smoke. There's no actual way to measure the amount that you're taking because you can't measure smoke, so therefore it can actually never be considered medicine. They do have CBD tinctures that are starting to become more widely available in gas stations and smoke shops. It is a legal gray area. It still is federally illegal. And again, there needs to be more research that's been done into it. There's been a lot of research that's been put out there, but it's mediocre. We need hardcore FDA research in order to move forward with conclusive evidence.
Thank you.
What would be the difference between a CBD derived from a hemp plant that you're seeing in a lot of the health food foods now and CBD derived from the cannabis plant?
Well, if you actually look, this is a great question. There is this whole indica sativa thing and how one plant makes you feel this and one plant makes you feel that. But if you actually look at the organic chemistry behind it, CBD, there's only one CBD molecule. You know what I mean? It's only one molecule to give you one effect. The rest of it's just marketing and a lot of misconceptions so that way it can keep you consuming. That's the business end of it. Again, it's primarily, for the most part, just smoke and mirrors. There's no actual scientific basis behind anything. CBD does one job. You know, THC does another job. That's it. Any other questions? No? Thank you very much.